Annually, the United States government spends a tremendous amount of money in biomedical research. Based upon this investment, it therefore is reasonable for taxpayers to ask, what did we get in return for that investment? To address this question, the Clinical Research Forum, a consortium of over 4 dozen leading academic health centers that conduct much of this research in humans, has put together a national competition entitled the Top 10 Clinical Research Accomplishments in the United States. What follows are those 10 accomplishments in summary, each of which is more exciting than the last. From curing leukemias that were previously uncurable, treating liver disease without any prior effective therapies, to marking disease in minority populations and developing biomarkers for early disease progression, these represent an enormous accomplishments from creative investigators all around the country. We hope you enjoy this presentation as much as we did in selecting them.
Emily relapsed in October 2011, and at that point, we knew it wasn't kind of the standard garden variety leukemia. Then she relapsed again in February 2012. And so it was at that point that we made the decision that we needed to go somewhere else where we knew that we needed to try something new and something different.
So we're really down to the CART-19 is what's left for Emily to cure her.
The essential nature of the CART-19 treatment is that we have to get cells from the patient. These cells are called T cells. They're cells of the immune system that need to be engineered. We can engineer these cells to go after cancer cells and kill them. About 3 weeks after we gave her her T cells, we did another bone marrow test. This young lady had not been in a remission for a number of months, and she now was in a complete remission.
We were able to go home June 1st. And since that time, she's been great. She's returned back to school.
We need to treat a larger number of patients before we understand what the success rate might actually be. Seems like the cells stick around for a long time. That actually might provide long-term disease control. That is, of course, the most exciting potential part. But we need more time to know if that's actually where we are. To see her go from leaving the hospital and starting to recover to basically going to school and playing soccer and looking like every other kid is just wonderful. I think it's the best thing about doing what I do.
Homozygous familial hypercholesterolemia, or HoFH, is a rare genetic disorder associated with extremely high levels of cholesterol, over 500 in most cases, and very early development of heart disease, often leading to heart attack and even death in children and adolescents and young adults. We work closely with a pharmaceutical company on the early studies of lamidopide, a novel way to lower cholesterol. And when that company decided for various reasons not to continue to develop it, I thought this would be a great way to potentially lower cholesterol in patients with homozygous FH. Through support from a foundation and ultimately from the federal government, we were able to do a critical proof of concept study that showed that lamidopide lowered cholesterol by about 50% in these patients, unheard of for this patient population, and really positioned us to actually further develop this with the hope of actually having it approved for treatment of cholesterol in these patients. In the pivotal trial, we treated patients for a year and a half and showed substantial reduction in cholesterol with acceptable safety and tolerability. And this study ultimately led to the approval by the FDA and the European Agency of lamidopide for treatment of patients with homozygous familial hypercholesterolemia. After more than a decade of research and development, it's gratifying that lamidopide is on the market, and for the first time, patients with homozygous FH have an effective treatment that substantially lowers their cholesterol levels. The availability of this new medication has energized the community of physicians who care for these patients and provided new hope for these patients with this devastating disease.
The REDUCE-MRSA trial was about keeping ICU patients safe from hospital-associated infections. This trial of 43 hospitals and 74 ICUs from Hospital Corporation of America found that bathing all patients with an antiseptic soap called chlorhexidine and using antibiotic ointment in the nose reduced a major antibiotic-resistant superbug by 37%. Even more impressively, it reduced all bloodstream infections by nearly half. This trial showed that there was an incredibly simple solution for preventing infections in our highest risk patients. It showed that treating all patients prevented more MRSA than identifying and targeting known MRSA carriers, suggesting that universal approaches have advantages. Finally, this collaboration is a shining example of how we learn together from everyday medical practice. This pragmatic trial had no study investigators on site. We provided protocols, education, and training, but the intervention was implemented by each hospital's own quality improvement infrastructure. This produced a generalizable trial for far less than the costs of randomized clinical trials.
My name is Denny Aberle. I am a professor of radiology and bioengineering at UCLA and a member of the UCLA Johnson Comprehensive Cancer Center. The work that I and my colleagues recently wrote on was relevant to the National Lung Screening Trial, which was a randomized trial that compared low-dose computed tomography, or CT, to chest X-ray as a screening tool. The paper that we wrote on was about not the baseline screen, but the 2 subsequent annual screens called incidence screens. In that study, we found that CT identified roughly 3 times more lung cancers than did chest X-ray. However, CT was also positive, meaning that the screen was felt to possibly relate to lung cancer, also roughly 3 times as often as chest X-ray. Overall, we observed a 20% relative difference in lung cancer deaths using CT relative to chest X-ray. The importance of this trial is that it is the first such trial that has shown a lung cancer screening tool to work effectively to reduce lung cancer mortality. We hope that with this intervention, we will be able to significantly negatively impact the number of individuals who die from lung cancer. Thank you.
The study essentially, in collaboration with Dr. Mundell, starts from very basic principles of identifying a specific molecule called B7-1 in the cells that are responsible for the filter barrier in the kidney, called podocytes. His lab, through many years of research, identified B7-1 as a molecule that causes injury to podocytes and leads to proteinuria kidney disease.
It was essential to partner with Dr. Greca because she does both research in the lab. She also has an active clinic.
We were able to give this drug called abatacept, which specifically blocks the target in the podocytes called B7-1. We gave it to patients with proteinuria kidney disease, and we were able to put them in remission for the first time. What
we have found is really the first targeted treatment for kidney disease. If confirmed in a clinical trial, this just changed the landscape.
We're very thankful that we're able to bring attention to this area where there's tremendous need for both children and adults suffering from these diseases, so that we can bring them new hope and new treatments and continue our very valuable work into the future.
For many years, the National Cancer Institute supported basic research. And we're at a time point now where all of this basic knowledge is coming forth with new therapies that target things specifically in cancer cells that allow for patient-friendly therapy. And the support coming from Congress for National Cancer Institute and other NIH awards to allow these new therapies to move forward to help patients is really essential for us to take the next step of making diseases like cancer, diseases that rather than patients suffering with side effects from therapy, changing it to a disease where patients can take patient-friendly therapies and live their lives without thinking about their disease. An exciting thing that we see happening now is many of these new therapies are coming forward. They're ready to be tested in patients, who have these devastating diseases. And really what we see is a lack of resources that with the cutbacks in federal funding has really impaired these new therapies moving forward. hopefully as we come out of the deep recession, we're going to see Congress appropriate more funds to facilitate these new therapies moving forward, which ultimately are going to bring down costs because patients with these diseases are going to be more productive, they're going to be able to work, they're going to be able to live their lives, and ultimately, contribute more to society in the future. In a perfect world, every analysis that I've seen relative to research, biomedical research and new therapies shows that every dollar that's invested brings back $6 to $7 in return, and that's not functioning in the patient quality, the patient quality, what they're going to contribute in the future. That's just what the investment brings back in terms of benefit to society.
Hello, my name is Larry Apple, and I'm giving this presentation on behalf of Linda Cow, who could not be here today. First of all, I want to thank the committee for selecting our research as one of the top 10 clinical research achievement awards for this year. We conducted 2 studies, one called ASK, African American Study of Kidney Disease and Hypertension, and the other called CRIC, Chronic Renal Insufficiency Cohort Study. These are 2 long-term observational studies sponsored by the National Institutes of Health. One of the most striking disparities in health is the extremely high risk of kidney disease and kidney failure in blacks compared to whites. Many people have thought this might be due to risk factors, but many of us also thought that there could be genetic causes. In these studies that we conducted, we identified a gene called ApoL1 that accelerated the progression of kidney disease in those African Americans who had the high-risk genetic variants. Interestingly, non-African Americans or whites don't have these genotypes, and so this genotype probably explains in part some of the striking disparities in kidney disease that we observe in the United States. A second important point I want to make is that these studies represent long-term investments by hundreds of scientists and staff that receive support from the NIH. We couldn't have done this without the support of the NIH. Thank you very much.
Warfarin is a drug that's been around for over 50 years and is used to prevent clotting in patients who are at risk of forming potentially life-threatening blood clots. Many patients require different doses, and trying to find the right dose for the right patient is a great challenge. If you give too much of the drug, you run the risk of bleeding, and if you give too little of the drug, it doesn't work. The COAG trial was a study sponsored by the National Institutes of Health, and it was designed to specifically test whether genetic information on top of what we know already about patients can provide benefit in selecting the right dose for patients beginning warfarin therapy. The trial demonstrated that the use of genetic information did not provide any additional benefit above and beyond what we can normally do with basic clinical information. In addition, there was a suggestion that in African Americans, clinical-based dosing was in fact better than dosing that added in genetic information. The COAG trial really demonstrates how important it is to do formal clinical trial testing in a rigorous fashion in order to really understand whether or not we should use genetics for dosing our patients or selecting drugs for those patients, particularly in complex situations such as warfarin.
More than 3 million people in the United States are living with hepatitis C virus, which causes chronic liver disease and leads to more deaths in the U.S. than HIV. Until now, treating hepatitis C required injections of a drug called interferon, which is very difficult for most patients to tolerate and requires up to 1 year of treatment. This paper begins a new chapter. Phase 3 clinical trials have demonstrated that new oral therapies for hepatitis C are shorter, simpler, more effective, and safer. As a result, the FDA approved the first interferon-free therapies for hepatitis C in December of 2013. Many people with hepatitis C unfortunately do not know they are infected. Because of the new screening guidelines for baby boomers, we expect to identify millions of these patients infected with hepatitis C in the next few years. It has been estimated that we can prevent more deaths in the United States, approaching 120,000, by linking hepatitis C screening to these improved treatment options. We wouldn't be celebrating this achievement without the public and private investments in hepatitis C research made by the National Institutes of Health and the pharmaceutical industry over the last 2 decades. This award honors the work of researchers and clinicians across the country and the thousands of patients who have volunteered to join us on this research journey.
This test, one of the main purpose is to reduce the need for a biopsy. You know, biopsies are safe these days, but still there are a lot of problems associated with them. And our test may help reduce the need for transplant biopsy, especially for the kidneys. So what is this test? We basically measure certain genes in the urine of the patient. So the patient, instead of undergoing a biopsy, just gives us about 50 cc's of urine. We spin it down in the lab, take cells from the urine and measure some genes. Many patients get the same amount of drugs, and these drugs are very useful in preventing transplant rejection, but sometimes they can cause infections and in the long term can lead to malignancy. Our test may help kind of personalize the therapy so the patient gets the right amount of drugs.
It's delightful for all of us to see the outstanding talent in clinical research and the wonderful work that's been done this year. The Clinical Research Forum strives to ensure that clinical research is supported, that it continues to contribute to the health of the public and the reduction of disease. I'm very proud to be associated with this marvelous effort and want to congratulate all the recipients of the awards this year.